Spinal muscular atrophy affects approximately 1 in 10,000 infants. Previously, SMA was a life-threatening illness, but thanks to the development of Zolgensma, children now have a chance at survival.
Spinal muscular atrophy is a genetic neuromuscular disorder caused by a mutation in the survival motor neuron 1 gene, resulting in the deficiency of the essential SMN protein. SMA is complex due to the presence of a highly similar gene called SMN2, which often produces less functional SMN protein. The number of SMN2 copies a person has can vary, influencing the disease's severity. SMN protein plays many crucial roles, including regulating protein levels in transcription and translation to forming small nuclear ribonucleoproteins involved in RNA splicing. Lacking the SMN protein results in interruption in many key cellular processes while disrupting protein homeostasis. SMA's hallmark is a selective motor neuron degeneration, leading to muscle weakness, atrophy, breathing difficulties, swallowing issues, and impaired motor function. The severity and progression of these symptoms depend on the specific SMA type and the available of the compensatory SMN proteins produced from SMN2.
Novartis researchers have developed Zolgensma. Zolgensma, active ingredient onasemnogene abe-parvovec, is a gene therapy used to treat spinal muscular atrophy in children under the age of 2. The drug consists of a recombinant self-complementary SMN gene enclosed by a viral vector, adeno-associated virus AAV9, which is used as a transportation method to deliver the drug. An IV is inserted into the patient's arm to insert Zolgensma into the bloodstream. This takes around an hour. Zolgensma travels through the bloodstream and eventually passes the blood-brain barrier, thanks to the AAV9 vector, to locate motor neuron cells in the central nervous system. Having reached these cells, it enters the nucleus and the viral vector is eventually discarded and expelled. The genetic sequence of Zolgensma exists as an episome, an independent piece of genetic material that is separate to the genome of the patient. The gene does not insert itself into the patient's DNA and is not inherited by their children. The gene is self-complementary, meaning it is ready to be transcribed once transcription factors bind to it. This produces SMN protein for the motor neurons of the patient.
The discovery of Zolgensma stems from research developed decades ago dating back to the 1960s. Slowly, as new information and research was published and put together, Zolgensma was produced. In the mid-1960s, Rowe, Hogan, and Atchison discovered the presence of adeno-associated viruses that can be genetically engineered to deliver and alter DNA in cells. This discovery of AAVs is what fueled the field of gene therapies. Fast forward to 2004, James Wilson's lab at the University of Pennsylvania discovered the AAV9 vector throughout their work to find gene therapy alternatives. They also discovered other AAVs such as AAV7 and AAV8, but found AAV9 to be extremely more effective at delivering genes to target cells. In 2009, Casper, Barkatz, and Azuz were effectively able to demonstrate that AAV9 is able to cross the blood-brain barrier in mice, further enabling the effective delivery of the gene to the target cells for SMA. Starting in 2010 all the way to 2019, clinical trials were underway in trying to prove that Zolgensma is an effective gene therapy for SMA. In May of 2019, Zolgensma received US FDA approval as a treatment for children under 2 years old.
Novartis operated a total of 7 clinical trials for the intravenous infusion of Zolgensma. The first Phase 1 clinical trial for Zolgensma, titled as START, was launched in April 2014, enrolling 15 patients with SMA type 1 who had 2 copies of the SMN2 backup gene and were 8 months of age or younger at the time of infusion. The goal for this trial was to establish safety and determine the appropriate dose of Zolgensma. Results of this clinical trial indicated that Zolgensma was effective at improving and sustaining muscle function, enabling patients to reach and maintain major motor milestones. At the end of this trial, 75% of patients could sit without support for at least 30 seconds, and 100% of patients survived without the need of permanent breathing support. Across all clinical trials, it was noted that Zolgensma poses a risk of hepatotoxicity where an increase of liver enzymes, including aminotransferase, results in liver failure. Recently, 2 cases of fatal acute liver failure occurred 6 to 7 weeks post-Zolgensma infusion.
In the world of gene therapy, it is crucial to strive for improvements that enhance both the clinical efficacy and cost efficiency of the drug. Some children, including those with SMA, have developed antibodies to AAV9, making them immune to its effects and unable to benefit from Zolgensma. One approach is to modify the AAV9 capsid, which acts as the outer shell of the virus used as a vector. This can happen through surface tethering, which is the attachment of specific molecules or ligands to the surface of the viral capsid, directing the modified virus to specific cells or tissues. This can also happen through encapsulation, which involves encapsulating genetic material within the viral capsid to target delivery of genetic material to host cells. Another approach is to attach antibodies that are specific to motor neuron receptors on the surface of AAV9 capsids, which significantly reduces the risk of interaction with non-target tissues. Finally, Zolgensma is a one-time treatment that comes with a staggering cost of $2.8 million per injection. Lowering the cost of this treatment is crucial for broader patient accessibility. By seeking solutions to these issues, muscles can keep working as they should. Zolgensma brings newfound hope to children battling SMA as it nurtures their strength and growth with each passing day.